And my contribution during that time was to show that besides AUG, which we know as an initiation codon, there is another codon, GUG, which can act as an initiator codon. Okay, so that's what I worked at the time. I later moved to Canada and still there. Now I would like to tell a few things during my time when I worked. First of all, in the first session, it seems that almost all these people who were speaking, they were in the lab or around the lab at that time. Okay, and it is very good to see them after 4 years. We had a similar meeting in Okayama University in Japan 4 years ago. Now, I came from California to Corana's lab in 66 January, very cold. And the thing is that I didn't have any top coat, so when I was in the airport, somebody told me, wait, your friends are getting you a top coat. And one of the friends is India who gave me the top coat, Dr. Agya. And then I came and went to the lab and Gobin asked what you want to work. I said, well, maybe this initiation problem. He said, okay. So I was in the lab, and the lab, I shared our first speaker, Dr. Wells, Bob Wells. Bob was in there, and always I'll come to Bob from his time. And then Dieter was in the other end of the lab, not in the same lab, but another lab on the other end of the hallway. And I was working also with Dieter, doing the ribosomal binding. So it was an exciting period. And one of the things is, what I remember very well, is that I was doing this initiation codon recognition, and for everybody else in the world who were working, like the people in Cambridge, MRC Boston, and not MRC Boston, Harvard, the biochemistry in Harvard, etc. So they could show the incorporation of amino acid as the truth, but not me. Gobin wanted everything to be established by an 8-1 degradation. And that's the time when we had the saying that 1 8-1 a day keeps Gobin away. And this 8-1 again, I never did any 8-1 degradation protein chemistry. Gobin said, well, I have done it. So don't worry. Okay, when he doesn't have time, he said, come on Saturday. So we'll do it on Saturday. So during that time I was greener. So Saturday morning coming around 9 o'clock was really tough. But still I managed to be there and go, you never shut up your results from Royal, I had G, I had this, that, and so he finally came up, and the first thing is that he opened my desk and the pipettes. The pipettes were, I had this way. He said, no, you keep the pipettes this way. Okay, so that's how he taught. So it was a great time, and without any further delay, I would like to start. And the first thing I would like to mention that being Canadian, I am following Canadian time, which is this hour has 50 minutes. So that means you guys now have 18 minutes. Okay? Okay, the first speaker is Professor Robert Wells, who has been for some time in Texas A&M University, Texas A&M, I think, Texas A&M University, and is in the Institute of Bioscience and Technology in Houston. Now Bob, as I said, Bob was in the lab, same lab as us, and he was in another side, on the other side of the bench, and on this side was my bench. And the thing is that Bob, when I would come, Bob's experiment is already done. We see him with his slide rule. Okay? And then he is planning for next experiment. Okay, he was one of these, but we could, and I never learned his management of time and his way. So Bob is going to talk about non-BDNA conformation, promote genomic rearrangement by defining the breakpoints. Bob.
Well, thank you very much, Laura, for that wonderful introduction. It's terrific to be back in Madison. It turns out that my wife and I spend summers here in Wisconsin because the summers in Houston, Texas are a little bit warmer than they are in Madison. We still have many old friends and associates here in the Madison area. It's great to be participating in the 33rd Steenbock Symposium. Interestingly, as I told Dr. Ansari, I organized Symposium number 2 in 1971 when I was a relatively young associate professor in the Department of Biochemistry here. So time really moves on, and this proves once again how time moves on. It's wonderful to be here to honor Goban. I guess he's going to join us in a little bit. But Goban really started me in the DNA field. I had been a peptide chemist and knew nothing about DNA. 1964, when I came to Madison to start my postdoc with Goban, we began working on the preparation of DNA polymers from defined repeating nucleotide sequences. Goban introduced me to DNA. That's an area that I have never left, even to this day, continuing to publish on DNA structural issues and the role of sequence in the properties of certain DNA segments. Interestingly, we have spent a good bit of our time identifying and characterizing different kinds of non-BDNA structures. But the underlying goal for the past 25 or more years has been, what do these things do? What are the functions of these non-BDNA structures? And I'll tell you about our efforts in this regard. So the first concept that I would like to get across is that DNA is polymorphic. And I think there is ample evidence. There are now thousands and thousands of papers, something like 25 million Google sites that describe this behavior. And that's a general theme of my presentation today. Some of the 11 non-BDNA structures that have been characterized are shown on this slide. Right-handed BDNA is the ordinary Watson-Crick form that we all see pictured time and time again, and in fact is on the cover of your book. But that's not all there is to it. There's more. At inverted repeat sequences, such as you see here, Watson can pair with Watson, and Crick will pair with Crick to form DNA cruciforms. It turns out that these structures are extremely important in the world of human genetics, as is being revealed by genomics as well as bioinformatics. DNA triplexes are three-stranded DNA structures that are formed at polypurine polypyrimidine runs, as shown here, where you have a three-stranded DNA structure, a region of small unpairing, and probably some unpairing of the fourth strand. Slip structures occur at direct repeat sequences, and the genomes, all genomes, are loaded with direct repeat sequences. So you can have folded back regions, as shown here, in the slip regions of the Watson strand and the Crick strand. Tetraplexes have been a tremendous growth industry in the past 10 years. And I was just at a meeting some months ago indicating the tremendous interest in DNA quadruplexes as shown here. They occur at G-rich regions. Left-handed zDNA occurring in alternating purine-pyrimidine sequences as you see here, where you have a left-handed region, and if you embed that in a right-handed segment of DNA, as shown on this slide, you must have VZ junctions. So you've really got some contortions. Sticky DNA has been shown to exist in the intron 1 of the Friedreich's ataxia gene. It occurs where you have GAA TTC repeats as shown here, namely very high bias of purines in one strand, pyrimidines in the other. And other conformational features are listed but not shown in detail, because as of this time they have not been shown to have the importance of the ones that I've shown here in genomic rearrangements. My laboratory, as I mentioned, has spent a great deal of time over the past 40 years in characterizing, identifying, understanding the factors that stabilize these structures, the interaction with ligands, small molecules, drugs, etc. And fortunately, we have been able to be the first to describe 7 of these 11 structures. But I'd like to turn my attention today to the topic of breakpoints, of gross deletions. Gross deletions are major genetic events that occur in a wide range of diseases that are the fundamental genetic basis for a number of diseases. And we first got interested in this in bacterial systems when investigating a very peculiar polypurine-polypyrimidine tract, 2.5 kb long, from the polycystic kidney disease gene. It turns out that if you have sequences which have a high propensity to form these non-BDNA structures, cells hate them. And whether it be bacterial cells, yeast, or human cells, they will all try to kick out these non-B structures. So what we did with the PKD gene, which in fact is 97% A's and G's in one strand. We spent a lot of time in the past several years in bioinformatics, have done lots and lots of searching, and this is one of the most unusual DNA sequences that we have found. It's 66% GC rich, but 97% of this 2.5 kb region has all A's and G's in one strand, T's and C's in the complementary strand. So it's a setup for forming triplexes and DNA split structures, and in fact, there are papers in the literature that describe this behavior. We looked at the products of the deletions and have sequenced now hundreds of these. And what we see is the following. If we look at mutant colon 1, we have first breakpoint and second breakpoint. So what is left is shown in capital letters. What is deleted is in lowercase. Deleted in lowercase, and what remains is in uppercase. So here's the junction region. So the breakpoint is right between these two T's and between this A and T to give this junction region. There are several interesting features, and now I will summarize this slide. There are several interesting features. One is that in virtually all cases there are small microhomologies, and they range from 1 base pair to about 15 base pairs. In general, I would say they are 3 to 10 base pairs in length. So here we have a CCTT, CCTT. Here's a little bit longer one. In some cases, it's as little as 1 base pair. So that's one interesting feature. Second interesting feature we asked is at these junctions, rather at the breakpoints which form the junctions, is there any propensity to form non-BDNA structures? And in virtually every case, in hundreds of cases that we have looked at, we can find non-BDNA structures, particularly cruciforms and SWIFT structures. So in the case that I just mentioned, where there is a deletion, several kb, which is called clone 4. In the vector region, you see these direct repeat sequences. The breakpoint is at the end of this arrow because that is what is deleted. So here's microhomology, and here is the microhomology in the polycystic kidney disease sequence. In the PKD sequence, there is this very stable triplex which is formed, and in the vector region, a DNA slip structure. And this is the rule, not the exception. We then decided to turn our attention away from bacterial systems and ask the question, what happens in human diseases? And very fortunately for us, David Cooper, University of Cardiff in Wales, has developed over the past 10 or so years a database, the Gross Rearrangement Breakpoint database, GROB-B, which has been extremely valuable. And I can't overemphasize the importance of David Cooper and his associates in this project. What we did is ask the question, is there a propensity to have alternating purine-pyrimidine sequences, which will form left-handed helices, and slip structures, or polypurine-polypyrimidines tracks, which will form triplexes, slip structures, etc. Is there a propensity for these tracks to be located near the human rearrangement breakpoints? We searched 125 base pairs on either side in 222 rearrangement cases, and I will show you the data from just some of those. But what you see is that the significance level in the distance to the nearest breakpoint, 5' or 3', for deletions and for translocations, is as shown on the right-hand side. Really very high level of significance. For the length sequences that you see here, 13 to 25, 5, 7, 11, or 7. So we believe that there is a substantial propensity for these sequences to be located near the breakpoints. Now on this and the next slide, I'm going to summarize approximately 30 papers. The repeat sequences that we have looked at are shown here. The myotonic dystrophy type 2 sequence, CCTG, CAGG, in the length shown here. Myotonic dystrophy type 1 sequence, the Friedreich's ataxia intron 1 sequence, the Fragile X sequence. But interestingly, this sequence, which I'll show you again later in another slide, has never been shown to form any non-DNA structure. Not in my lab, nor in any other lab. The types of rearrangements that we find are gross deletions, and these are the prominent forms. But we also see inversions, we see insertions, we see copy number variations, and a wide variety of complex rearrangements. What I mean by complex rearrangements is anything more complicated than these, i.e. generally a combination of 2 or more other types of rearrangements. The systems in general that we've looked at are bacterial systems and several human cells, and the results to summarize in this body of work. First, rearrangements of up to about 4 kb have been found, and this is now some 700 or 800 cases that we looked at. Non-B structures at all the breakpoints, particularly slip structures, cruciforms, tetraplexes, and triplexes. But the most important point I think I would like to convey is that the recombined junctions contain remnants of the non-BDNA structures. That is to say, ladies and gentlemen, what we see in the starting materials are what we see in the products from the rearrangement events are remnants. Namely, we see a cruciform that is cut at the non-paired regions. We see triplexes that are cut at the non-paired regions. So it's as if there has been a footprint left by the non-BDNA structures. For us, this was a very important discovery. It was important because, just coming back to this last point, it was important because my lab over the in the 1970s, 80s, and early 90s, published a number of papers, as did virtually all the other labs in the field, published a number of papers looking at the influence of left-handed zDNA or a triplex on various events, transcription, recombination, DNA replication, et cetera, et cetera. And yes, there are a lot of papers in the literature, and yes, people worked hard on this, but I don't, as far as I'm concerned, it was never very convincing. And it wasn't convincing because there was no obligatory role. In this case, there is obviously an obligatory role of the non-vDNA structures. Other workers in my lab a couple of years ago showed that non-vDNA conformations, which are formed by long repeating tracks, not the sequences per se, are promoting the mutagenesis in flanking regions. In other words, it's the conformations, not the sequences per se. How did we do that? We did it by 3 different techniques, and I will describe 2 of these. One is from in vitro experiments. We knew very well that there is an important influence of negative supercoiled density on the propensity to form a left-handed Z structure or a cruciform or a triplex. We did the same experiments inside living cells, E. coli, in either GYR-B mutants, which lower the supercoiled density, or top A mutants, which in fact increase the negative supercoiled density, put in more supercoils. And what we found is that if you decrease the supercoiled density, you decrease mutagenesis. If you increase supercoiled density, you increase mutagenesis, just as you would expect. Another line of evidence is that we looked at sequences which either do or do not form non-vDNA structures. I've already mentioned that CAA-TTC repeats do not form any type of non-vDNA structure, and neither my lab nor any other has found any mutagenic behavior. However, these 4 sequences in different lengths will form different types of non-vDNA structures, and in fact are very strongly mutagenic in the type of assays that I just mentioned. And these are just some other non-vDNA structures which have been characterized. What you see here is a slip structure with a slip structure, and here's the deleted region. Here is another slip structure with a cruciform, and so on. We then turned our attention to the deletion breakpoints in certain human diseases, and we looked very hard at 11 diseases. They are listed here, and I would like to just point out a couple of these. One is the azoospermia factor that has been investigated so beautifully by David Page and his associates in Boston. I've already mentioned autosomal dominant polycystic kidney disease, but let me mention adrenoleukodystrophy because I'm going to show you the sequence of this in just a second. The deletion of 4Kb in the adrenoleukodystrophy gene is shown here. The types of non-vDNA structures that can be found are a DNA quadruplex on one side. Here is the microhomology, this GG, and here with the GG some 4Kb away. This forms a very nice cruciform, but there are even other types of non-V structures that can be formed. In fact, between a non-paired region associated with this quadruplex and, in fact, this oligopurine, oligopyrimidine-rich region. Let me give you an overview that is written up in this Tibbs article that I published a couple of years ago, or I guess, yes, a year and a half ago, on the concept that we believe is operative. First, in the chromosome, DNA segments exist either in the right-handed B form or in non-B conformations at specific loci. Small direct or inverted homologies give rise to recognition of the weakened DNA structures at or near the breakpoints. And through double-strand break mechanisms and recombination repair processes, you have a DNA joining and healing. which will give rise to gross deletions, translocations, copy number variations, inversions, insertions, or duplications, and through multiple steps in pathology give rise to a variety of genomic disorders. What are these genomic disorders? Well, there are about 70 now that have been characterized by the world in the world of human genetics. And let me say, ladies and gentlemen, that our work has absolutely taken off in the past 6 or 7 years due to the involvement of genomics, bioinformatics, human genetics, and medicine combined with biochemistry and molecular biology. In this Tibbs article, I have listed and have discussed these different diseases. Some of these are familiar to some of you. I mentioned the spermatogenetic failure, which causes a giant deletion due to a very large cruciform. But in fact, David Page is carefully identified at an inverted repeat region. I might also point out chronic myeloid leukemia with an inverted repeat, and there are a number of other diseases that beyond those that in fact I've listed here. So let me summarize simply by saying that non-DNA conformations promote rearrangements by defining breakpoints, obviously meaning that non-DNA conformations are the basis of human disease. I don't think for a minute this is all the non-DNA structures do, but it's a start. We've been very fortunate to collaborate with a terrific group of other workers, not only in my institute, but at other institutions. Thank you very much.